I'm here today in front of the ENSTBB, a French institute of biotechnology specialized in the purification, characterization, and production of biomolecules. These schools offer the 3 years training, which associates different needs in the biotech field, such as biotechnology and bioproduction, engineering, and business administration. Today, we're going to talk about how and why to use bacteria for protein production. Proteins used to be extracted from organisms, but the increasing demand of the market and the safety of this process incited pharmaceuticals companies to find alternative ways of production. Today, we will only focus on one alternative, which is the production of recombinant protein using bacteria. The advantage of this kind of process is that it's safer, more profitable, and renewable. Furthermore, thanks to advanced technologies such as molecular biology and bioproduction, we can both improve the strain and the properties of protein. Hi, Nicolas.
Hi.
You're a third-year student at the ENSTBB. Could you give us a quick overview of how to produce protein using bacteria?
Yeah, of course. So, first you need to understand that cells continually produce protein from genes. The production of a protein using bacterium can be split into 3 main stages. The first one is molecular biology, which corresponds to the introduction of the gene of interest into the bacteria. Then, the bacteria is put under specific conditions in order to grow well and produce the protein of interest, but in addition to its own. These steps so far correspond to the upstream part of the process. Then we use separation methods to isolate and purify the protein of interest. And these correspond to the downstream part of the process.
Thank you, Nicolas. So today, we will only focus on the upstream part of the process.
The first step is the molecular biology part. The goal is to insert the gene encoding for the protein of interest into a circular DNA molecule called the plasmid. This plasmid is then inserted into bacteria, which replicates the plasmids when duplicating. The plasmids also contain a gene encoding a protein, giving the bacteria an antibiotic resistance. This trick allows bacteria to be selected using a medium containing this antibiotic. After having selected only the bacteria carrying the target plasmid, the production process can now begin.
Before starting protein production in bioreactor, a starter culture has to be prepared. In order to do this, bacteria selected in the previous step are grown in a small volume of medium, about 10% of the final volume. This allows bacteria to adapt to the medium and to the culture parameters such as temperature and stirring. When their growth rate is at its maximum, all the preculture is transferred to the bioreactor, which is pre-filled with the same growth medium, about 90% of the final volume. So let's get more information about the bioreactor with Laurent, my colleague at ENSTBB. Hi Laurent, can you describe the system please?
Hi, you can see there a 5 liters bioreactor which can be inoculated with preculture. You can find in this system the reactor tank, which will contain the culture and suspension, an agitation system, different pump, feeding pump, and 2 others for the addition of base or acid. Thermal jackets for the temperature control, an air supply system, and finally you can see there an entry and an exit for sampling or harvesting.
Okay, thank you Laurent.
In fact, the bioreactor is able to reproduce the living condition of cells. The main parameters that have to be controlled in the bioreactor are the aeration, which brings oxygen to cells, the agitation, which allows cells to stay in suspension and distribute the oxygen in the bioreactor evenly. The pH and temperature are important parameters to control because each strain has its optimal pH and temperature. Metabolites produced during the culture make the pH fluctuate, and it is regulated by the addition of acid or base. The temperature is controlled by the thermal jacket that surrounds the bioreactor. Feeding is carried out thanks to a pump.
Among all the parameters to control, production parameters are critical. To realize these measurements, samples are regularly taken from the culture. It's essential to measure biomass formation by measuring optical density using a spectral photometer. Substrate consumption also has to be monitored to ensure that cells are correctly fed. And last but not least, the production of the protein of interest is the most important setting to monitor.
The final step of an upstream process and the first step of a downstream process is to harvest the protein of interest. A centrifugation step is performed to separate the bacteria from the medium. If the protein are secreted, the purification steps are directly based on the medium. If the proteins are intracellular, the bacteria have to be lysed, the lysate clarified to remove the cell debris, and the purification is finally performed on the clarified lysate. After several purification steps, a pure solution of the protein of interest is obtained.
To summarize, we saw today that producing proteins using bacteria must be carefully monitored to ensure an optimal cell growth. Once the process is well controlled, the next challenge is the scale-up. Two steps are needed before commercial production. The first one is the optimization of the process using a pilot bioreactor, and then an industrial one. Now, if you want any further information, please visit www.enstbb.ipb.fr. Thank you.